Look at this glowing waveform. This rapidly updating trace provides a direct, real-time window into the specific rhythm and mechanical strength of a human heart. This is an electrocardiogram, or ECG. It is a diagnostic test that records the electrical activity of the heart over a set period of time, capturing exactly how the heart beats. The ECG functions as a precise spatiotemporal map. It tracks the physical location of electrical signals as they travel through the cardiac tissue and measures the strict timing of the resulting mechanical contractions. By recording these signals, the ECG translates invisible physiological actions into a globally readable, standardized diagnostic waveform. Every heartbeat begins with a biological trigger. The mechanical contraction and relaxation of cardiac muscle is driven entirely by preceding electrical impulses generated within the heart itself. These impulses are quantified using Ohm's law, V equals I times R. In this biological context, V is the measured voltage, I is the electrical current generated by the heart muscles, and R is the electrical resistance provided by human body tissues. Because human tissue offers resistance, the currents reaching the surface are incredibly faint. An ECG machine operates as a sensitive amplifier, detecting these microsignals across the skin and magnifying them for analysis. The resulting clinical graph is an exercise in pure physics, a direct plotting of physiological voltage against time. The cycle starts in the right atrium, near the entrance of the superior vena cava. Here, the sinoatrial node, or SA node, acts as the natural pacemaker, firing the initial spark that dictates the heart rate. In this split-screen visualization, watch the left side as electrical impulses spread from the SA node throughout both atria, stimulating them to depolarize and contract. On the right, this event draws the first small curve on the ECG grid, the P wave, lasting between 0.08 and 0.10 seconds. The signal then reaches the atrioventricular, or AV node, located near the AV valve. The AV node acts as an electrical gateway, deliberately delaying the passage of the impulse. On the graph, this pause traces a flat horizontal line, known as the PR interval, lasting roughly 0.12 to 0.20 seconds. This electrical delay ensures the atria have completely ejected all their blood into the lower ventricles before the next phase of the cycle begins. This initial fraction of the ECG trace provides clinical proof that the upper chambers have successfully primed the primary pumps. Once released from the AV node, the signal passes into the AV bundle, or bundle of His. It divides into left and right branches, shooting toward the apex of the heart, before spreading upward through the Purkinje fibers. On the graph, this ventricular depolarization draws the massive spike of the QRS complex. This complex typically lasts 0.06 to 0.10 seconds and consists of 3 parts. The Q wave tracks the depolarization of the central interventricular septum. The towering R wave is produced by the firing of the main ventricular mass, and the S wave represents the final phase of depolarization at the base of the heart. During this exact window, the atria are also repolarizing and resetting. However, the electrical voltage generated by the ventricles is so large that it obscures the atrial signal on the graph. The QRS complex visually quantifies the electrical force required to power the thick ventricular walls and pump blood out to the entire body. Following the spike, the graph plots a flat line called the ST segment. This reflects a plateau in the myocardial action potential, representing the duration when the ventricles are actively contracting and pumping blood. Immediately after, the trace curves upward into the T wave. This represents ventricular repolarization, the electrical reset that occurs right before the ventricles relax during diastole. We can measure the total combined time required for all ventricular electrical activity, from initial depolarization to final repolarization. This is the QT interval, normally lasting between 0.35 and 0.44 seconds. This completed PQRST sequence provides a mathematical snapshot of a single healthy cardiac cycle. To capture this data, technicians attach electrodes to specific points on patients' chest, arms, and mics. These leads connect to the ECG machine, which records the electrical signals for several seconds. Accurate data relies on precise setup. Technicians must verify machine calibration, ensure firm electrode contact, and require the patient to remain completely still and silent, as even minor movements can introduce interference. This recording occurs across 4 primary environments: standard resting ECGs, physical stress ECGs to assess the heart under load, continuous Holter monitoring via portable devices, and real-time telemetry for critical hospital patients. Flawless physical protocols and appropriate hardware selection are mandatory. Any data artifact can mimic the presentation of a fatal cardiac event on the final readout. The diagnostic power of the ECG lies in baseline comparison. Physicians identify specific deviations from the standard PQRST durations, intervals, and wave shapes. Specific abnormalities point directly to structural failures. Because damaged tissue conducts electricity differently than healthy tissue, wave distortions can pinpoint tissue ischemia, or the physical location of a myocardial infarction. Chaotic or irregular rhythms allow doctors to diagnose electrical failures, such as arrhythmias or dangerous electrolyte imbalances that interfere with cellular depolarization. By tracking the timing and shape of the electrical current, the ECG serves as a frontline triage tool capable of pinpointing the nature of a cardiac failure. This diagnostic precision extends into pharmaceutical research. During clinical trials for new systemic medications, such as antihypertensive drugs, continuous ECG monitoring is a mandatory safety requirement. Look at this stacked comparison. Researchers are monitoring for drug-induced QT prolongation. The top line shows a standard heartbeat, while the bottom reveals a visibly stretched QT interval, indicating a delayed electrical reset of the ventricles. This prolonged interval is a primary indicator of cardiac risk. This monitoring identifies potential complications early, preventing the progression toward lethal drug side effects. The ECG provides high-resolution data on heart function through a simple, non-invasive procedure. By translating fundamental physics into actionable clinical data, the ECG remains the primary standard for patient safety, drug evaluation, and clinical decision-making.